Okay, so again, I want to insist on the principle that it's a Q&A. So if I say something you don't understand, just ask questions, don't hesitate, even in the middle of a sentence, just scream and say, hey, I don't understand that. Could you repeat or explain in a better way? Okay, so I'm going to share. Okay, let's start. Need to move that. Um, is it okay? Did you see properly? Everything is fine? Okay, great. So today's subject is how do we know if a treatment works? So, in fact, last month I explained the what's the even 2 months ago, what's the basic of the of Myhre syndrome. That's all the different manifestations that we have. That is developmental, progressive, multisystemic, which means that we have different system, nervous system, gastrointestinal system, and etc. And it's ultra rare. It's a monogenic, which means that there is only one gene that is affected. It's autosomal dominant, so it means that only one of the two genes, either from the mother or the father, needs to have a mutation to, in fact, express the disease. And we have only a few mutations that triggers Myhre, mainly the I500V or the R496C. So you might recognize the mutation of your kids here. And so the mutation is in the gene SMAD4, and it's at the end of the TGF beta superfamily. So that was if you want to understand better, you were not there last month, just go back on the YouTube channel and you will have more explanations about that. So today I really want to explain you the principle of drug discovery, how to really measure that, and the fundamentals, in fact, how to do that. So for MSF, for the foundation, we are focusing a lot of our time on drug discovery. We want to find a treatment, but our fundamentals is that the patient is really at the center. So we have the patient priorities at the center. And at the same time, we want to create tools, because if you want to test something, you need to have the tools. We want to give access to those tools to as many researchers over the world and people who want to test medicine in Myhre. We want to have a balanced portfolio. So the portfolio of treatment, I talked about that last month. So the different type of treatment that we are developing now for Myhre. I talked about that last month. And what is really important is to align with the regulation, because if you test drugs, discover new treatments, but you need to have the authorization from the FDA in the US, the EMA in Europe, but you cannot just give medication like that to patients. So what's very important is that the FDA and the EMA agree on the way on your tools, agree on the way you are going to measure the efficiency of a treatment. We set the priorities, as I explained before, according to the patients. The areas of priority were cardiovascular, respiratory, and behavioral. And on a mechanistic point of view, it's fibrosis and stenosis. So today I'm going to talk about the tools, really. What do we need and what did we develop at the foundation as tools to test treatments? So there are two large phases in developing a treatment. You have what we call the preclinical phase, which is before testing in humans. Obviously, when you discover a new treatment, you are not going to test it directly in patients. That's a bit dangerous. So you first test it before going on human, you test it what we call in vitro, which means at the cellular levels. So you work on cells. And then in vivo, which means you test the drugs on or the treatment on whole organisms, on living animals. And then when you have shown that it's efficient in vitro and in vivo and that it's not toxic, then you ask the authorities, the FDA, please look at that. It's fantastic. It's not toxic. Could we test it on patient? And that's what we call the clinical phases. So that testing in human, then you do again toxicity. If you have a large population and a lot of money, you do toxicity in healthy volunteers. In rare disease, we tend to have this phase combined with the efficacy in patients. So usually it's toxicity and then efficacy in patients. But you can imagine that to do that, you need different things. So I'm going to focus now in preclinical phase, and then I will talk about the clinical phase. So in the preclinical phase, the first thing you do is that you need to identify the target. You need to know what is the problem. So fortunately, that's great. For Myhre, we know what is the problem. The team of Valérie Cormédière identified the mutations in the SMAD4 gene associated with the Myhre syndrome. That was already years and years ago. In the past few years, several teams around the world identified that these mutations trigger an overstabilization of the SMAD complex that I explained last time, which means a gain of function. So it's not just one scientist who discovered that. We can be confident in that conclusion because we have the team from Souley Mo at Emory University, Hongmei Mu at Harvard, Maria Macias in Barcelona, Spain, and the team of Park in South Korea. They all arrived at the same conclusion that the problem with the mutation of SMAD4 is that there is an overstabilization of the SMAD complex. So now we know the origin of the problem, we know what the problem is. So what we want to determine is what to modify to decrease or remove the problem. So we know that it's a gene mutation, so you can correct the mutation. That's really the highest level you can do. We know it's an overstabilization of the complex, so you can try to destabilize the complex. You can also decrease the trigger of SMAD4 activation, because SMAD4, I told you before, is at the end of the whole TGF-beta family that triggers the activation of SMAD4. You can try to remove this activation so that SMAD4 is not on. And another level is that you can act on the symptoms, which would be the consequences of SMAD4 gain of function. So those are different levels. So now, when you choose where you are going to act as a treatment, you need to select the method to get this modification. So, for example, if you want to correct the mutation, that's gene editing. If you want to destabilize the complex, you can use small molecules. That's the traditional medication that usually everybody takes. That's what we call small molecules. You can use immunotherapy to destroy the consequences. So what I explained last time that we are developing a FAP CAR T project, treatment, which is in fact using your own immune system to destroy the cells that are overreacting in fibrosis. So that could be also another way to act on the consequences of SMAD4 gain of function, which would be, for example, the fibrosis. So then when you have that, you know the methods, you're going to test if the modification work. So how do we do that? So test the modification. Does it work? First, you test on what? You test on something that will represent the disease. For example, at the different level, that can be, you can test on SMAD4 gene DNA. am I able to correct the mutation? You are going to test on the gene. If you want to destabilize the complex, you can isolate the protein SMAD4 and work directly on the SMAD4 mutated protein. You can work on at the cellular level, just cells containing the mutated SMAD4, or you can work on organoids. So you cannot take the organs from a patient, but you can recreate organoids, so similar to an organ that contains the mutated SMAD4, or you can test it on a whole organism, which could be the animals with the SMAD4 mutation. And what we do is that we try to have, well, I'll explain that later. So a whole animal. When you know on what you are going to measure, you need to do what to measure. So something that the disease modifies. So for example, the integrity of the gene, DNA. The level of SMAD complex stabilization. Does it stabilize a lot, a little bit? So that's what you can measure. You can, if you are at the cellular level, you are going to measure the normal, typical cell function. The cell that has SMAD4 mutation doesn't work properly. You put the treatment on it, and then you look at the cells. Does it function properly or not? Is the function better than the one without treatment? Then you can also in organoids, if you look at organoids model, you can look at the development of the organ with the treatment. Does it develop better and does it function better? And when you test a treatment on a whole organism, on a living animal, then you can look at the reduction or the disappearance, let's hope, of the Myhre syndrome phenotype. So the way you see the disease appear in the animal. So when you have that, it means that you have models, so that's on what you test, and you have assays. So the assays are really the technology you use to measure the efficacy of a treatment. Do you have any questions? Do you understand how to test more or less? Yeah, no questions?
Yeah. This might be the wrong time to ask a question, but when you talked about gene editing, small molecules, and immunotherapy, is there an order or path to least resistance when it comes to either of those? Like, are we going to go, because I know losartan obviously had the pilot clinical trial. Like, are there easier and quicker ways to go with some of this, or is it just we need to look at what's going to be the most effective as the first point of reference?
Well, we don't know. That's the whole problem with the drug discovery or treatment discovery is that as long as you are not testing the treatment, you don't know which one is going to work.
Hmm.
So that's the reason why we say that we develop a balanced portfolio. So we have more innovative technology, more risky technology, really safe technologies like the Fab CAR T, for example, is used on hundred thousands of people everywhere in the world. So we know that it's safe. So that's where we put our money is really on Fab CAR T to reduce the fibrosis. But at the same time, we invest in gene editing, and at the same time, we invest in small molecules, and we test all of that in parallel, and we don't know which one is going to
Which,
be.
yeah. Do the FDA and the European regulatory body, uh, is it easier to go down a small molecule route, or is that just an oversimplification? Because obviously, with some of the technologies, there's kind of a no going back kind of situation with some of them. Whereas with small molecules, obviously, it can be in a weekly or daily tablet form of which you can monitor dosage and all that kind of thing. Is that like an oversimplification of the situation?
No, not really. You're right that gene editing is way more difficult to get approved by the FDA than small molecules. Although small molecules is not easy either, because
Mhm.
the problem with small molecules is that you have less specificity of the treatment, so more side effects, and they are
Mhm.
very be careful with the side effects. The immunotherapy is really doesn't show any problem at the regulation level because it's not toxic or
Mm,
very limited toxicity compared to small molecules. And it's proven to be a technology that works. So yeah, the FDA is not neutral in front of the different type of treatments. Um, but still, I mean, if we work really closely in collaboration with the FDA and developing the things with them, giving some advice to us on how to progress in the discovery of treatments, then we know that we will have, if it works, of course. If it doesn't work, we won't have the agreement. But if we do everything by the rules and in very strict following the requirements of the FDA, we have a big chance, if the treatment works, to have the authorization to be approved.
mm,
So that's there's some tricks and requirements that we need to follow, and that's what we are doing
mm.
in
Cool, great. Thank you.
our things. Thanks, Kate. Is there any other remarks or questions? Because not everyone was there last month, so it's true that the different treatment that we are testing might not be, people might not be aware of what we do. Okay, so let's go on with the preclinical tools. So to test the modification, if it works or not, you have models, so on what to test, and that's the models I just mentioned. And then you have assays with the different things that you are going to measure. There is a level of complexity in the different models and in the different assays. So if you start from the genes and you go to whole organisms, so animal, living animal, then you have the degree of complexity that increases a lot. You can imagine that a whole animal is more complex than a piece of DNA in a bacteria. And it's also the throughput. So the number of molecules you can test is way different. If you put cells, you can have 1 million cells in a petri dish that big. You cannot have 1 million mice as easily as 1 million cell. So if you want to test, as we are going to do next month, 800 molecules in Myhre syndrome, you are not going to test 800 molecules in mice, because then you need thousands of mice, and that would take 10 years. So what you do is that you test first on a model that is less complex. So first, those molecules were first tested directly on the protein, on the mutated protein. They tested 260,000 molecules on the protein. And from those 260,000, they selected 800 that are efficient in destabilizing the complex. So now we are going to test them at the cellular level. And from those 800 at the cellular level, we might end up with 10 that are going to be brought in a more complex, and maybe one or two in a whole organism, in the mice. Um, so what you are going to test at the same time, when you test at the cellular level, you are going to test two things, maybe two different functions of the cell. You are going to test in maybe two type of cells. We chose to look at the brain endothelial cells, so that makes the vascular vascularization in the brain. So we test on those cells, and we test on microglial, which are also in the brain, for the reactivity of the brain. Then we go further in more complicated models, and at the end, we'll test in the mice, we'll test in many different things, on the fibrosis that is shown in the mice, on the, I'm going to talk better with the information, in fact, on the behavioral, the sleep. So the level of complexity is very important, and there is a way to go. You don't start with mice, you start with cells. Except if you have one molecule and you want to know about one molecule or one treatment. But usually you test a lot and then you reduce up to the more complex system. So we were saying that we have to choose a model. So how did we choose the models? We want to be as close as we can to patient's phenotype. Phenotype means the manifestation, the way the patient is different from non-sick person. So we want to be close to the patients. We want to avoid animal source to build the models because we have been doing that in drug discovery for the past 40 years. And we have a lot of molecules to treat mice, but very few to treat humans because mice are not humans. So what we did is that we created iPSCs, so it means inducible pluripotent stem cells. So we took the blood from the patients, we isolated the cells from the blood, and then put them back at the level of stem cells. So those are not stem cells, they are cells from the blood. We put them back at the level of stem cells and then reprogram them in the cells we want. So in brain cells, in vascular cells. So that we have, in fact, a model that is really the cells from the patients, but we didn't have to go in their brain to take a piece of brain or to cut a piece of vessels or artery. We can recreate liver, we can recreate a lot of different organs from the cells from the blood from the patients. At the end, we need to have a whole animal. So what we did is that we are creating, it's not yet ready. We have other scientists that created animal models for the I500V mutation, and we are creating, that should be ready in February, the animal model for the R496C mutation, and those models are humanized, which means that we took the gene from the human gene and we put it in the mice. We are not working on the mice genes, we are working on human genes in the mice that we put in the mice. So in front of that, what to measure? So how do you choose what you are going to measure? First, you need to, so you have models. First, you need to characterize and understand the models. So if you have, for example, the protein as a model, you see that Maria Macias, for example, in Spain, really did a very nice 3D model of the protein so that she can understand, she's a chemist, so that she can understand where her molecules are going to interact in the protein. At the foundation, we created a preclinical platform starting from at the cellular level. So with the iPSCs that I just mentioned from the patients. So we have characterized those cells. We understand now a lot of things for the past year. Thanks to this model that we could study and characterize, we could understand a lot of mechanism of the Myhre mutation that comes from the Myhre mutation. It's gave us a lot of information. Then when you have really characterized, you understand what's different between the cell with the mutation, with the Myhre mutation, and the cells with the, in fact, we took non-affected siblings of the same sex and approximately same age, and we could compare those two behavior of the cells. And then we selected, okay, that's really significant. This behavior of the cell is really significant to Myhre, so proof that there is Myhre syndrome. So we are going to test on that function on that characteristic in the cell. Then we optimize the technology. What are the conditions to measure? We do what we call an assay optimization and validation. And from there, we check with the FDA, is the assay good? Do you agree that what we are doing is really relevant, that it's standardized, and that it's going to be significant if we show that a treatment is modifying what we are measuring, the characteristic we are measuring? That's a tricky concept. Do you have any questions on that concept? No? Oh, all clear? Maybe not interested. So that's what we did. That's in the past year. Well, the organoids are not ready yet. But what we did in the past year is really construct those tools, preclinical tools, and that's what we call the MSF preclinical platform. So we have the cells, and that's really the cells from a Myhre patient. It's not a picture that I took anywhere. That's the cells from the I500V. Yeah, that's the I500V, and that, whoops, sorry. Okay. So that's the patient-derived iPSC reprogram, and those are the brain microvascular endothelial cells. And we have 3D vascularization, so which means that we can really recreate the network of vessels in a petri dish. There is written in big NAMs. So NAMs means non-animal models. So those models that we chose, and I insist on that because the FDA and the authority and the people that are really testing, doing drug discovery, insist now that we should go away from those models derived from animals. We should go closer and closer to patients and to models that are really human models, non-animal models, human and close to the patients. So that's the way we developed our preclinical platform. I will jump directly. We are planning the organoids, but I mean we need funds for that, and we need to set priorities. So the priorities we had were the cells and the mouse model, but we plan to get some funding so that we would be able to have blood vessel organoid and skin organoid. The skin organoid would be a good way to test treatment because this is a characteristic that is really over a majority of Myhre patients. So we have also the humanized mice model. I just said that that's the R496C will be available in February. We are doing what we call phenotypic assays. So that means that we study the mice and we study their respiratory, musculoskeletal characteristics, their behavioral characteristics, how do they sleep, do they socialize or not, are they anxious, and we even have an assay that is based on vocalization. So are they speaking later? I mean, speaking as mice can speak, but are they verbally communicating earlier, later? So we measure all these things. We take some samples and measure the fibrosis also, so that we can have a baseline and then test the treatments in the mice and see if this baseline is modified and improved. Do you have any questions here?
We're going to ask a really annoying question, which I know is not easy to answer. So, timing. And I know you've mentioned some of these things obviously have not been started because you need funding. Like, so what, obviously that is, I love this because it's such a great way to show like the progress towards clinical trial. But what stage are we at in terms of being able to say, okay, our target is X amount of years, or this typically takes this long, or is it all just dependent on a lot of things?
Well, no, it's not I mean, it's a rather it's a very good questions, and I think for part of the portfolio we can answer quite easily. For the IPS, so the cellular level, the in vitro, we are going to do the cellular assay in a month. Exactly. So we should have the results in a few months later, not a month, but maybe 6 months later, which means that at the same time, we should characterize the mice, and this could go for the best compound directly in the mice. And when we are in the mice, then it takes a bit longer, and what's the takes quite money and time is the PKPD. So that's the last line you see on here, where in fact it's the toxicity study. So to see what's the highest dose that we can put in the mice, the is it toxic, where does it go in the body? So that's, I mean, that should be that's a question of a year maybe. So now what's very important is the type of small molecules that we are going to test in the cellular in the cells next month. Some
Mm.
of those are what we call NCEs, so new chemical entities. So they are new drugs. So if you start from a new drug, you have to do all the PKPD toxicity. It's a lot of work, and it's really long. It's 2, 3 years. If you in those 800 compounds, there are also repurposing drugs, which means drugs that are already on the market, meaning that all the toxicology studies has been done already, and it has been proven that those are relatively toxic or non-toxic for humans. That since they are on the market, we know that they are okay to be on the market. So those would go way faster, would go maybe after a year, you can be already going to humans. So the molecule you start from will determine, in fact, the length of the study, the time it will take. Now, if you go for the FAP-CAR-T program that we started a few months ago, is meant to be in human, if it works, of course, in about 3 years. But that's for fibrosis.
Yeah.
Uh,
Well, it's very exciting. Gosh,
yeah,
that's
yeah.
huge.
And for the gene editing, that would be a bit longer. But for the gene editing, we tested briefly on the if the technology works on cells, and we are going to start immediately in mice models because we constructed a specific, very specific mouse model that allow us to do screening of gene editing directly in the mouse. It's a very different from the other type of mouse model that we have.
Mhm. And sorry, I'll be quiet in a second. The repurposing drugs.
No, please no.
You'll be like, oh God, I don't want her to come to anymore. So for the repurposing, when they've already had, obviously, they're a step forward in toxicity and testing, and so there's a comfort level there from a sign off. Is it that we're actually creating new mixtures of those compounds? So it might be 20% of something, or is it actually just a purely relabeled drug that we would then literally repurpose a drug that might be used for something else and then get permission to use it for this?
No, that's not a mix of molecules. Usually it's only one molecule.
Just one. Okay.
Just one. But what we are talking with, in fact, different companies, and it's confidential, so I cannot say which
Of
one.
course, yeah.
But we are talking to a company that would give the compound. They're already testing in humans, not for Myhre, but
Hmm.
they would be with Myhre, and they have different compounds. In fact, another one that is at the level of mouse model, that would be in what we call combination therapy with the FAP CAR T.
Ah, okay.
So that's an option. And since we are talking about that, I'm now um, contacting other pharmaceutical companies for some other compounds or molecules that are in clinical trials now, not for Myhre, but I'm trying to convince them to include
Mm.
Myhre patients, and especially Myhre patients that have tracheal stenosis. So the issue there is not medical or scientific issue, it's a legal issue
Mm.
because the companies they buy from the chemist. Usually, it's a chemist that makes a compound, so they buy from the chemist the license, so the right to use those molecules. So they buy the molecules, but they buy them usually not for everything. They have to precise what they want to use it for. So, for example, there is a molecule that could work for tracheal stenosis for us. I mean, there are several that could work, but one of them, in fact, was sold to a company for solid tumor and cancer only. So those people cannot give us the molecule or the treatments because we are not part of cancer therapy. We are part of fibrosis, rather behavioral fibrosis things. So we have to go to the other company that bought the molecule for the application of fibrosis, behavioral, or whatever. So that complicates a lot our work, but we are doing it. And there is not only one company, but several company and several molecules, treatments, potential treatments for Myhre that are already in clinical trials for other applications that we are pursuing and trying to convince them to include our patients. So when we talk to them, the fact that we have exactly that slide, the MSF preclinical platform, is a good way to convince them. They are very happy. When they see that, they know that they can have the preclinical data required by the FDA to agree for a clinical trial. So that's extremely important, and that's the reason why the foundation for the past year really focused on that, so that we can convince the company that we have the tools to get the data to the FDA to convince them that it's not toxic for the patient, that we are not going to kill the patient with it, that it's working, that it would be beneficial for the patient, so that they would agree that we use those molecules. I'm talking.
Fabulous.
I don't
No,
know.
no, that's
I
awesome.
answer a question. I don't even remember your
No,
question.
no, no. And I think that shows, isn't it? This platform is just making that decision make for them so much easier because they're satisfied that this you are ticking the boxes that need to be ticked and that you can take on some of that with them in partnership. That's amazing. Great, thank you.
So now going to the clinicals. So not the left, the right parts. We go to human. Do you have any questions on the before humans? Because now I'm going to move to humans.
I do. I'm sorry. This is Mary Porter.
Yeah.
So my question is, and I don't know if you can see me. I don't know if I
Yeah.
started my video. Okay. So I'm a hospital pharmacist, and so I'm kind of familiar with immunotherapy a little bit. I know you can't tell us the names of the drugs or anything that they're looking at, but can you tell us like therapeutic class? Are we looking at like monoclonal antibodies or
No,
what kind?
yeah, good questions. So when I say immunotherapy, it's cellular therapy. So it's the CAR T, that they use in the
Okay.
cancer. So
Yep.
it's your T cells, so your
Uh-huh.
immune system that is
Yeah.
modified to really targets certain cells, and the
Okay.
modification we are doing, in fact, they are doing at the Geneton in Paris and with Valérie Cormier at the Imagine Institute. They are looking at modifying the T cells from the patients so that they will recognize the fibrotic tissue and be able to destroy all the fibrotic tissue that you have everywhere.
Okay.
Triggers the joint issues, the respiratory, cardiac, and eventually stenosis of the trachea. So
Okay.
that's really using your T cells, your immune
Yeah.
system, to
Yeah.
attack and
Interesting.
destroy. That was first developed years and years ago for cancer. So
Uh-huh.
they train the T cells from the patients to recognize the cancer cells so that they will destroy the cancer cells.
Okay.
So
Okay.
the Geneton, Isabel Richards, sent us a proposal saying we want to destroy the fibrosis in Myhre patients. What do you think? And we, since it was already half developed for Duchenne myopathy, they have fibrosis in their muscles. They were already quite advanced in the project. At the foundation, we said, okay, let's go for that. Let's try to destroy the fibrosis in the patient using this CAR T technology, which is proven to be efficient,
Hmm.
not proven with the fibrosis yet. That's new, but the CAR T in itself is proven. So that's
Hmm.
that's the immunotherapy we're actively working on.
Okay. Thank you very much. Sorry if you said that earlier and I missed it. So,
Well,
um,
that
th-
was last month. You can go to the where I explained really the Fab CAR
sure.
T technology. But I mean, I
Yeah.
understand that everybody cannot be every week at
Yeah.
the
Yeah.
every month at the thing. So that's the reason why we put it
Sure.
on YouTube, and I'm very happy to explain
Yeah.
um, and answer all your questions.
Yeah.
Really great.
I'll look that one up. Thank you very much for
You're welcome.
clarifying.
So now we have tested our treatments on cells in the animals. We see, well, there is a good treatment that works in the patient cells, in the animals. It's not toxic. We did the PK/PD, so we know exactly, well, not exactly because it's on an animal, but we have an idea about the dosage, about the toxicity, about the efficiency. So we can go to humans. I mean, the FDA decides if we can go to humans. Now we are going to go through the same things, test the toxicity, and test if it's efficient in patients. So the model, again, it's like the preclinical. You will need models. So you will say, well, it's in humans. So the models, they are patients. It's not that easy because we have uh, a disease that is heterogeneous in the manifestation and progressive. So which patient do you choose? So you have to prioritize and focus on specific manifestation where maybe you will have to separate and do subpopulation of patients. So for us, that's not ideal because we have already something like 200 patients. So if we have to do small subpopulation of 10 patients, then statistically, when you have results, that becomes really difficult. So that's not really the way we would like to go. Um, and it's a progressive disease. So do we have to separate the population, adults versus pediatric population? So those are all the things that we are working on now with the clinicians from clinicians from everywhere, from Australia to Europe to US. Everybody is involved, and we are all we are in a working group that we organize at the foundation, trying to answer those questions. Um, so then you need the assays. You remember on cells or animal model, you need to know what you are going to measure. So in human, it's called clinical outcome measure. So what to measure? Well, Again, it has to be the same, something that the disease modifies. So we need to characterize and understand the models, so the patients. We need to select one or several of those characteristics and measure in a reproducible way. So the way you measure in London is going to be the same as in Sydney in Australia and the same as in New York. I live in New York. So that's very important. If you want to compare the results, you need to be really standardized. Everybody does it the same way, measure the same way. And if you measure every 2 months or 3 months, it has also to be exactly the same way. And it has to be a technology that is really tailored and adapted to Myhre. You can take some type of measurement that are applied already in other diseases, but you need to be very careful that it's exactly tailored to Myhre syndrome. That's very important because there are so many rare diseases that had good treatments, and the treatment was refused, not approved by the FDA or the EMA because the measure they were doing, the clinical outcome they chose, was not really exactly relevant to their disease. So the efficiency of the treatment, although the patient could say that my life is changed, it's really great, the measure, the way they measured and the characteristic they measured was not showing the difference, was not tailored, adapted to their disease, and the drug was rejected. So that's something extremely important for us, is that we need to have exactly the right measurement for us. And then we check with the FDA, do you agree with that? Do you agree with this type of measurement? So what we need to do to characterize and understand the model, usually it's called a longitudinal natural history study. So it means that you follow the evolution of the patients by measuring relevant outcome and quantify the progression of the disease. So we didn't really do that up to now. So you need to establish a baseline in different subpopulation of the Myhre syndrome population in some cases, and other cases, other characteristic that you need to measure in the whole population. So that's a bit complicated. So we're working on that also. Then you select, when you have really the characteristic, you select the most relevant manifestation to measure, so the clinical outcome, and then the method to measure it. That's very important. So that's exactly what I was saying before. If there is no method that is really tailored to Myhre syndrome, that we need to create new tools and optimize the technology. So that's something we are also working on, or we'll start working on, because we just had the few first meetings. So we'll see but that's the idea, to create the new tools. And then again, check with the FDA or the EMA if the assays are good enough to draw reliable conclusions, if they are happy with the way we do things. Because at the end, they are the ones to decide. So what we do with the working group of clinicians that we have gathered is that we work on, since we are so heterogeneous in the manifestation and you have patients that are really different, we chose domains, and we are going to develop some clinical trials, not all of them, but some clinical trials with what we call a composite multi-domain endpoint. It means that we choose 4 different domains, and we are going to make a kind of ratio and normalize the data so that we can integrate all these different points and see the global action of a treatment. So the domains we chose were the skin and joint, because it's standardized, so we know exactly how to do it. It's very precise. It can be done with the same tool. It's very specific tool everywhere in all the clinic centers that are going to do the clinical trial. So that's very standardized. There are already some data from non-Myhre syndrome patients, I mean, not patients, from the general population. So we can really compare to the general population and we can compare the evolution of the same patient. Does the regulatory agree on it? Yes, it was already included in several clinical trials in the past. So the FDA and the EMA and the Asian authority are totally okay with this type of measurements. So that's good. So we know exactly what we are going to do. Now, the second domain is cardiac and respiratory. So that's really where we have not a consensus. We don't really know what we are going to use. There are a lot of discussions among the clinicians on that. Because they are not really adapted to the fact that we are pediatric, the fact that we have autism and ADHD, and it's a mix of different type of populations. So what we are doing now, we are talking to companies to try to develop a tool that would summarize and integrate, not summarize, that would integrate all those different characteristics in only one. It's a wearable in one wearable that would be tested in clinics and then at home, so that we could measure also in the real world, in the everyday life, if the treatment has a modification and has an action or not on the patient. The third domain is gastrointestinal. So we know exactly what we are going to measure. It's different surveys that the patient has to fill in. So it's standardized, of course, very easy, cheap, which is also something we need to think about because clinical trials cost a lot of money, several $1 million usually per clinical trial. So we need to include things that are going to be okay financially for us also. The last domain is behavioral and cognitive, and that will be the clinical outcome assessment will be in sleep and in different behavioral surveys. So there is written here, I said about clinical outcome assessments, but there is endpoint. So endpoint is in fact the term we use to say, this is exactly what I want to measure to see if the treatment is efficient in the patient. So in traditional clinical trials, which are not composite multi-domain endpoints, you choose one primary endpoint, then you choose a secondary endpoint and exploratory endpoints. For us, since we are very heterogeneous, we need to have a multi-domain endpoint. So really, when we decide, for example, for cancer, it's easy. You put your endpoint as 20% reduction of a tumor. That's the endpoint. For us, we are still working on the exact definition of the endpoint. So we need to determine for the skin and joints at what angle we will say that the treatment is efficient or not. For the cardiac and respiratory, where we are developing the tool, but for the gastrointestinal, we need also to determine the endpoint, meaning at what number of spontaneous bowel movement per week are we going to say that the treatment is efficient? So those are the endpoints that we are working on. So that's it's not done, it's not finished. We hope that it will be all finished in about a year from now. And we would be able to start baseline studies. So not yet well, if we can start a clinical trial, that's great. But at least we could start with the baseline studies. There is one now clinical trial conducted on losartan in Paris by Valérie Cormédière. That's the only clinical trial with Myhre, the official clinical trial with Myhre syndrome now. We hope that talking to the different companies will have access to some more drugs and maybe could participate to clinical trials with other diseases that are in common with common manifestation as us. So that's what we are working on. Do you have any questions? Oh, I did that in an hour. Okay.
Hi, this is Karen Rich. Sorry, this is my first time attending, and I have a very basic question. So I understand what the drugs, part of what part of what you would be testing is behavioral cognitive, and the drugs are meant to treat the fibrosis. And I've never understood or been clear on whether the belief is that the behavioral cognitive symptoms are caused by fibrosis or not. And so I was curious if that's a known thing or not.
No, so we don't really know. We don't, when we do MRI of the patient, we don't really see any type of fibrosis. There are a few issues with microvascular, brain microvascular in some patients. But the thing is, SMAD4, I'm a neuroscientist, so I can tell you that SMAD4 is really known in neuroscience because SMAD is at the center of neurodevelopment. So to develop the brain, SMAD is extremely important, and you need the protocol that everybody uses to develop iPSCs into neurons is what is called dual in SMAD inhibitors. So everybody uses the inhibition of SMAD to trigger neurons. So imagine the patients that have a SMAD4 gain of function. So they have difficulties developing the brain. And that's the reason why there is this behavioral and cognitive issues. It's really developmental. Then after, as adults, SMAD4 in the nervous system is still very important. You have in the brain, you have neurons, but you have the majority of the cells are not neurons. They are what we call glial cells. So astrocytes, microglia, or other type. I'm not going to give you all the details, but the astrocytes, for example, the proliferation in the diencephalon, which is the central part of the brain, depends on SMAD4. So those cells that are the cells that are going to clean the brain and help the neurons, support the neurons, they depend on SMAD4. So that's not fibrosis. That could be, in a way, if you make a parallel between the nervous system and the peripheral system, you could say that the reactivity of glial cells might be in a way compared to fibrosis. That's a stretch, but I mean, if you're a neuroscientist, you can say, okay, those are reactive cells that will make a scar in the brain. So you can compare that to fibrosis in the rest in the skin, for example, or in the trachea, or that will react as an over wound healing that will make the scar. Well, in the brain, those are the astrocytes and the microglial cells. So even if SMAD4 is fundamental for the development of the brain, it's also in adults it's very important in the glial cells, so the support for the scarring and the reaction to aggression to the brain. Sorry, I talk a lot, so you need to stop me when you say, okay, I understand. When you want me to stop, just say.
Yeah, no, that makes sense. So, but the treatments that you're looking at, the drugs you're looking at, the thought is that they would also target those cognitive issues as well.
Yeah, so if they can penetrate the brain, of course, because that's an issue with small molecules, is that not all of them can pass the blood-brain barrier. But if they can penetrate the brain, those you're talking about the small molecules that we are going to test next month. Yeah. So those are destabilizing the SMAD4 complex. So they are really they are not at the gene, but they are very close to the very root of the cause of the disease. So if you can have regulate SMAD4 activity to a rather normal activity, then it will act on everything, on all the symptoms that you have with Myhre syndrome, with the condition that the drugs penetrate the brain, of course.
Thank you.
That's the reason why we really focus on that, and we put that in priorities. That it's more a priority for us to develop something that would work on SMAD4 than the drugs that would work on the TGF beta, which is at more general level and less specific to Myhre. But we still go talk to companies for the more general TGF beta inhibitors or antagonists. We try to work on every level, all the different level that we can, but the results are going to be different depending on the type of treatment we go for.